studying about foodborne infection and intoxication in today's class. Now, what is this what is food poisoning? Now, this is illness caused due to food that is contaminated with bacteria, viruses, parasites, or if it has been contaminated with their toxins. Maybe the microorganisms are not there, these bacteria and fungi, viruses, they are not there, but it could be that they had released out their toxins into these food substances, and those food substances that we are consuming have the toxins produced by them. So that is called as food poisoning. Now it can be categorized into two types, that is infection and intoxication. Now let us see what is this infection and intoxication. Infection is a process where the food itself, the food has the present microorganisms, that is microorganisms are physically present in the food that we are consuming, and those organisms enter into our system and they cause different illness. Okay, so that is infection directly by the microorganism. What is intoxication? Intoxication, as I told you in the previous one, that is the microorganisms may not be there in the food, may or may not be there in the food, but we suffer from illness, okay, because of the toxins that is released by the microorganisms or that is present in the food. Okay, so that's called as intoxication. So we can have some various examples of food poisoning. That is intoxication, that is your Staphylococcus aureus, Bacillus. The very good example is Clostridium botulinum, Clostridium perfringens is a very good example of toxin, food intoxication. And food infection is your Salmonella, E.coli, Shigella, Listeria, all these come under food infection. This doesn't mean that they do not produce toxin. They could produce toxin, but their mere presence of the microorganism is responsible for the disease that you have. But here, it's not necessary that microorganisms should be present in toxin, but the toxin itself will result in intoxication. Coming to the first organism that you have in your syllabus, that is Escherichia coli. If we have to talk about Escherichia coli, we have to start with the classification. It comes under domain bacteria, phylum Proteobacteria, order Enterobacteriales, family Enterobacteriaceae, genus Escherichia, species coli. Now, why this Enterobacteriaceae? Entero means what? Inside, okay. All the organisms, all the bacteria that is present in the intestine or cause infection in the intestine, present or can cause infection in the intestine, in the colon. So that is why all those organisms come under this group, this family Enterobacteriaceae. Entero meaning inside. Okay. So this is the classification. Now let us see the morphology of this organism. I think it was first studied and explained by Theodor Escherich in the year 1885. So that is why on behalf of him they have named this organism as Escherichia coli, because it was Escherich who had studied about this organism. You all know that this is a gram-negative organism, rod shape. They are short rods. They have very short rods, and hence they could be confused as cocci also. They could be also called as cocobacilli because they are not big rods. They are short rods, but they are not cocci. So they can be also called as cocobacillary form. They are aerobic in nature. They are opportunistic pathogens. Now, what do you mean by opportunistic pathogens? See, these organisms, Escherichia coli, they are present in your intestine. They have to be present in your intestine. They are the normal flora of the intestine. But when your immune system reduces, when they find an opportunity, they could be pathogenic for your body. Okay, so that's called as opportunistic pathogens. These organisms also could revert to that situation. So that's what I told you, they are the normal flora of the lower intestine in warm-blooded organisms. Moving on to, if we grow these organisms in the media, how do they look? Because this is very, very essential for us to understand to know how the organism look on media, because for detection. On nutrient agar, the colonies are small, pasty, and off-white colonies are seen. On McConkey's agar, they look like this, that is, they show pink colored colonies. Now, what do you mean by pink colored colonies on McConkey's agar? Which means they can ferment lactose. They are lactose fermenting organism. Okay, and one special media for this is EMB, that is eosin methylene blue agar, wherein these organisms, that is Escherichia, E. coli, if E. coli grows on EMB, it will produce a green metallic sheen. You can see here, green metallic sheen colonies are. So if it is E. coli, you can find green metallic sheen. Other organisms, if it is Enterobacter or any other organism, they may produce any other color, pink colony or black colony, but it is not green metallic sheen. So if on EMB, if you see green metallic sheen, means you can confirm it as E. coli. Coming to the next concept, that is how does it spread? How does it get contaminated? As I told, it is present in the intestine of warm-blooded animals. So definitely it will be spread through, it is shed out through the fecal matter in animals and also in human beings. So if it spreads through feco-oral route. Now what does this feco-oral route means? Now it means that it spreads, it gets released out in the feces, as I told you. And if the person practices unhygienic conditions, that is, the person does not wash the hand properly after using the toilet, and same hand uses for cooking food, the easily the organism can get mixed into the food. That is one thing. Also, it can spread through fomites, inanimate objects. Now what do you mean by fomites? Again, if the person has used the toilet and does not wash the hand properly, and with the same hand he has used the definitely handle of the toilet, and the other person also has washed the hand properly but then uses this hand to open the handle, the same handle, the organism gets transferred onto him and the organism enters into his body. Without washing his hands he may eat. So that's how. Direct contact, that is through shaking of the hand, etc. It spreads through water also. Now what do you, how it spreads through water? That is, if the drinking water gets contaminated with the sewage water, easily spreads through water. It can be present in food, that is, it can be present in vegetables, fruits grown. How? That is, if the fruits and vegetables grown, if the farm, the water that is used in farming, if that is contaminated with E. coli, or if they are adding any manure that is contaminated with E. coli, definitely the food, the vegetables and the fruits, the greens, the green leafy vegetables easily can get contaminated by this organism. So this is how it spreads. Coming to the symptoms. Now what does it mainly cause? It causes food poisoning, that is gastrointestinal diseases. Now what does it cause? It causes diarrhea, dysentery, abdominal pain. What is the difference between diarrhea and dysentery? That is, diarrhea is loose watery stool, dysentery along with mucus and blood. Obviously it is a foreign particle that is entering into your system, so you get fever. There is dehydration. Excess of dehydration is seen because there is loss of water. Diarrhea means loose stools, excess loss of water is there. Obviously you are losing the salts from your body, so there is weakness, lethargy, and there is loss of appetite seen. Apart from this, there is also UTI. The people can also suffer from urinary tract infection because of these organisms. See the bloody stains to urine. Now coming to its pathogenicity. Now how exactly this organism causes the disease? See, any organism to cause a disease, it can be either through the toxin production or because of the antigenic structures it has. Now if any organism do not have either of this, anything of this, then they cannot cause a disease. Okay, so now this organism, they have antigenic structures also, it has toxins also, both it has. Let us see what are the antigenic structures it has. It has O antigen, it is called as a somatic O antigen. It serves as endotoxic activity. It has H antigen, that is, this H antigen is also called as flagellar H antigen. This is present on the flagella. Now it contributes for the protein fimbriae antigen, okay, and it has K antigen which protects, that is capsular K antigen it is called as, it protects phagocytosis, that is, it protects the organism from being phagocytized, being engulfed by the macrophages, because it is slippery, the capsule is slippery in nature. Capsular K antigen it is called as. Some of them, they also have this VI, virulence VI antigen. So this antigen is present on the surface of the organism. Now let us see what are the toxins that is present. It has LT and ST toxin. Now what is this LT and ST toxin? LT is heat labile toxin and ST is heat stable toxin. As the name says, LT easily gets disintegrated when during high temperature. Heat stable, it can resist higher temperatures. Okay, now what is this toxin? If you can see this diagram here, it is made up of 1A subunit surrounded by 5B subunit. It's basically protein units, protein fragments. Okay, so 1A subunit fragment and surrounded by 5B subunit fragments. Now this A as such is it is not active. It does not get activated until and unless this A gets split up into two fragments. Okay, so let us see how does it get split up into two fragments. This B subunit helps the entire toxin to get bound to the surface of the intestine. Okay, so that's why it is called as binding. Basically, it helps in binding. So as this, when this toxin gets an opportunity to bind, it has receptors so that it can bind to the surface of the intestine. So when it gets bound, what happens? Automatically this B subunit gets disintegrated and this A subunits get split into two, that is A1 and A2. Now what this A1 and A2 will do is it will increase the activity of CAMP, adenosine monophosphate. What that will do is it will create an imbalance, ion imbalance what this ion imbalance will do is this, it will increase the adenylase cyclase activity, because of which there will be excess accumulation, entire your intestine, or this toxin pulls the water content into the intestine from your body. It pulls the water content into your intestine. As a result, your intestine is now filled with water, excess of water. Your intestine cannot hold the water, and thus resulting in diarrhea. So this is how it causes the disease. Okay, now there are different phenotypes of this organism, that is your ETEC, EPEC, EHEC and EAEC. Now let's see what is this. ETEC is called as enterotoxigenic E. coli. Please remember it is enterotoxigenic E. coli. This causes traveler's diarrhea. Now what is the meaning of this traveler's diarrhea is, you would have seen that people who are traveling from, you can say, the developed countries, when they are traveling to the developing countries, they will end up in getting this diarrhea like symptoms. Now what is this? The people, those who are in the developing countries, they maybe they're consuming the organism E. coli may be present in smaller amounts in water or in the food. So your body does not recognize this as a foreign substance. It's got used to it. But when those people, developed country people, they are always used to drinking, pure water, mineral water, which do not have any microorganism. So when they drink the water, when they come here and drink the water, definitely their body starts reacting to it by producing these kind of symptoms, diarrhea like symptoms. That's ETEC generally causes diarrhea like symptoms, that is watery diarrhea, but that is without fear. Okay, you can see this in human beings. And the next one that you see is EPEC, that is enteropathogenic E. coli. This causes infection in infants, that is in children, basically between the age 6 months, okay, 0 to 6 months of age. Also it can be seen, that is diarrhea. And you can also see this infection in cats, dogs, rabbits, etc. Then the next one, EHEC. Now this is generally, H means enterohemorrhagic. From the name itself it tells hemorrhage. So what this organism will do is, it will go and sit on your intestine lining. It will invade into the intestine. It will cause a hemorrhage there. Okay, as a result it will start eating up the mucus. It will cause hemorrhage. It will cause inflammation in that area. Okay, so it causes diarrhea and dysentery along with inflammation. Because of inflammation, definitely dysentery. I told you what is the meaning of dysentery in the previous slide. It causes infection in human, cattle and goats. EHEC, remember. Now what is this EAEC? Now this is enteroaggregative E. coli. Now what this is, this organism goes and sits in one place in your intestine. It aggregates, it colonizes there. Okay, it generally causes infection in human beings only. Okay, and also it can lyse the cell. Basically it does not invade into the cell. Okay, it cannot invade, it cannot, but it can lyse the cell, and generally causes infection only in human beings. Now you get here watery diarrhea without fever. So these are the different phenotypic types of Escherichia. What are the foods that is generally associated? Basically it could be any kind of food if the person is unhygienically handling the food. As I had told you, person uses the washroom and toilet, and without washing the hand comes and cooks the food, any food can get easily contaminated. Otherwise, ground beef, unpasteurized milk and milk products, fruits, green leafy vegetables. I had told you how the fruits and green leafy vegetables can get contaminated if the farm uses the water that is contaminated by E. coli. Diagnosis would be, what are the samples that you can check for? Generally the stool sample is taken to check if, definitely if the person is infected, he will shed out these kind of different types of E. coli. So stool sample can be taken, the water sample can be taken, the suspected food sample can be taken. What is done? Gram staining is done, culture testing is done to check the presence of this organism. Prevention. Now this organism, as you have seen, they do not have any kind of resistance, that is they do not have spores, endospores, anything as such. So which means if you cook or if you heat, organism easily gets killed. So thorough cooking of the raw food from any animal source will easily kill the microorganism. Thoroughly washing of vegetables, because these organisms hardly penetrate inside, so is washing of the vegetables and also cooking the vegetables thoroughly before eating. Avoid drinking of unpasteurized milk or milk products. So pasteurization of milk is a better choice. Maintaining hygienic conditions. Okay, even if you cook the food properly, wash the vegetables, but your hands itself are contaminated, you're having the food, definitely you can get the organism. Okay, so these are the preventive measures generally that you can have. Treatment, as this organism generally causes uh, loss of water, oral rehydration, that is continuous supply of water, and salts has to be given. Antibiotics not recommended because usually within 1 to 7 days you'll be fine. But if it becomes chronic, amoxicillin and cephalosporins could be given. Thank you.